Okay, so let's start the class. In the previous class, we started to study about the antibody, and we said that the antibody is a glycoprotein which first recognize the antigen, and after the recognitions, they bind over the antigen and facilitate the antigen clearance in our body. When I am using the word facilitate, it means either antibody खुद मार देता है एंटीजन को, और else ये एंटीजन को लेके किसी दूसरे इम्यून सेल के पास जाता है, प्रेजेंट करता है. फाइन. दूसरे जो मोर पावरफुल सेल्स है लाइक द मैक्रोफेजेस, मोनोसाइट्स, उसके पास लेके जाता है कि अब तुम मेरे से नहीं हो रहा, तुम इसको मारो. बट रिकॉग्नाइजेशन करने, पहचान करने की जो कैपेबिलिटी है वो एंटीबॉडी के पास होता है. Then we discussed that 30% पूरे ब्लड में अगर हम देखते हैं तो इतना परसेंटेज केवल एंटीबॉडी है. एंटीबॉडी प्राइमरली बी लिम्फोसाइट मींस बोन मैरो के अंदर में ये प्रोड्यूस्ड होते हैं, और ये हमारे सीरम पार्ट ऑफ द ब्लड जिसमें कि हम लोग क्लॉटिंग प्रोटीन को हटा देते हैं, तो जो ब्लड का पार्ट बचता है उसे हम लोग सीरम कहते हैं. वो सीरम के किस पार्ट में है? प्लाज्मा में है. प्लाज्मा के ग्लोब्यूलिन एंटीबॉडीज बनाते हैं. देन वी डिस्कस्ड अबाउट देयर स्ट्रक्चर. स्ट्रक्चरली दे आर टेट्रापेप्टाइड, 2 लाइट चेन प्लस 2 heavy chain joined by the disulfide bond, and there is a specific area called as antigen binding site जहाँ पे एंटीजन बाइंड करते हैं. Apart from that, we will also discuss that based on physiology and antigenic differences, antibodies are 5 classes. Five classes. We can easily remember by remembering the mnemonics called GAMED, IgG, IgA, IgM, IgE, and IgD. Then we also discussed that some of the antibodies, they are alone, that is called as the monomers. We discussed that some of the antibodies, they join by the another antibody through the J chain, and that is called as the dimeric form. Fine. One antibody can bind with the 2 antigen. So valency, if you're talking about the valency, then monomeric form of antibody have the divalent valency. Then if antibody is a dimeric, then valency is tetra, 4 valency. Then we discussed that antibody can exist in the form of pentamer. Pentamer means J chain having the 5 antibody joined. Okay, and the valency is 10. Then we discussed that in the antibody there is some area that is a constant. The lower portion of antibody is constant and upper portion is a variable because it have to vary according to the antigenic shape, because antigens are diverse in shape. Okay, this much we discussed. Now in today's class we are going bit depth to understanding each of the antibodies, So let's write first according to the concentrations. We developed the one mnemonics called as the GAMED. Yes. Now, so in the GAMED we will start with the antibody G. See, this mnemonics also helps you to remember their concentration in the body. G is the most abundant. Fine. Second most abundant antibody is IgA. Then IgM, E or D is found in the traces, a very low amount in traces. Fine. Now starting with the IgA antibody. Antibody is represented as A, capital A, small b. It is antibody. Okay. So we're discussing the antibody IgG. Okay. Yeah, it is means antibody. Antibody IgG. The first thing you have to understand is it is the most abundant. सबसे ज़्यादा पाया जाता है. Among all the antibodies, this is the most abundant. Okay. Most abundant. Second thing which you have to understand is why it is most abundant. The reason is Why it is most abundant? The reason being the half-life. Half-life means how much this antibody can survive in our body, how much days basically. That is called as the half-life. So half-life of antibody IgG is 23 days. Is 23 days. Fine. And this half-life is जितना भी अभी हम लोग एंटीबॉडी पढ़ने वाले हैं, सबसे ज़्यादा जीने वाला एंटीबॉडी यही है, 23 days. The second thing which you can say more give the favor why this is most abundant is, like suppose I got an infection. Infections by means of any bacterial infections or any viral infection, anything. Take the example of bacteria, and you know that the part which basically is pathogenic in the bacteria is called as antigen. It's called as the antigens. So antigen comes, fine. Antigens comes. When first time antigen comes, then there will be antibody will produce, no? Yes. In the presence of antigen, our body synthesize the antibody. जब कभी एंटीजन आता है तो हम लोग एंटीबॉडी बनाते हैं. इट्स लाइक गेस्ट हमारे घर में आता है तो हम अच्छा खाना बना के उनको खिलाते हैं. फाइन. बट हियर द गेस्ट इज इनेमी. हियर द गेस्ट इज इनेमी. सो द थिंग इज व्हेन एंटीजन कम्स, then our body synthesize the antibody. Now these antibody which is synthesized in the first, you can say, interaction, in the first exposure, is IgG. So IgG antibody, it is a first immune response antibody. Write it down. It is a, it is a first immune response antibody. Clear? And the thing is the first exposure. It is a first exposure, also called as the primary immune response. Also called as the primary immune response. Which antibody produced is the IgG. But when there will be a subsequent, or you can say the secondary exposure to the same infections, then there is a production of another antibody. Yes, and that is called as, this antibody is called as IgM. Fine. Why it's happened? The reason is, when first time antigen comes, then IgG antibody starts secreting in our body. IgM is in a less amount. But when there will be a second, then first in initial exposure, memory is formed about that antigen. पहला एक्सपोजर में जो बैक्टीरिया और वायरस का एंटीजन आया था, उसके खिलाफ हमारे बॉडी ने मेमोरी सेल्स डेवलप कर लिया. एंड आफ्टर द सेकेंड एक्सपोजर, this memory cell will switch this IgG into the IgM. So there is a IgM outburst, large amount, fine. And IgG in a slowly decline. No problem. Now another thing which you have to remember about the IgG is that this exists in a monomeric form. Now you have the clear idea, no? What is meant by the monomer? So this is the, monomeric form. It exists as a monomeric form. Clear? No problem? Yes? Fine. Another thing, yes? You want weight? Weight of antibody, it is 150 Dalton. See, the weight is not here important. The thing which you have to understand is the function. And how it's produced in our body, why it is synthesized in our body, and their half-life. Now, so it's a 150 Dalton in weight. Now, talking about there, you can say, the IgG antibody, this is the only one antibody which you are getting from your mother, mother. When you are in your intrauterine life period, when you are in a intrauterine means when you are in a fetus form inside your mother uterus, then there is a one link, okay, which basically connect with your mother, that is called as the placenta. Fine. So through the placenta there will be a nutrition you are getting, and there will be a gaseous exchange. Fine. So from that placenta, mother is giving you the antibody called as the IgG, and this IgG antibody protects you in your initial period of life. So this is the, it can cross the placenta. It can cross the placenta. Clear? So transplacentally you are getting the IgG antibody. Clear? Now, the another function which is done by the IgG antibody is its help in the phagocytosis. Now what I mean by the phagocytosis? Phagocytosis says that, once again, phagocytosis means engulfing. It means engulfing. In our body, there are cells called as the macrophages, neutrophils. They basically engulf the antigen. So what happened? When any antigen comes, suppose this is the bacterial antigen, then antibody will bind over here. Yes, antibody binds. Now, one side, this is which region? Binding region, and this region is binded with the antigen. Fine. Then this part, okay, this part, it will bind with the macrophages. Macrophages have the receptor of this antibody, that is called as the IgG. Fine. And it bind with the macrophage. And when it's bind, macrophage knows that कि कोई antigen आया है. What I said, what is called as the definition of antibody. I said that antibodies is a glycoprotein. Fine. ये पहचानता है antigen को. After पहचानना, what it does, it basically facilitation means either कि वो खुद मार देगा या फिर किसी दूसरे हायर मॉलिक्यूल्स को इन्फॉर्म करेगा कि there is some antigen, act like antigen presenting cells. Okay, ये एंटीजन प्रेजेंट करेगा दूसरे सेल्स के पास, जो मोर पावरफुल सेल्स है उसके पास. So what happened? This antigen, it is presented to the macrophages, and now macrophage what will do? It will engulf. Fine. So you can see it like. So this is the engulfed part of antigen. Clear? This process is called as the opsonization. This is called as the write down. It's maybe two marks they will ask. This is called as the opsonization. So basically, opsonization is the process by which antibody one part will bind with the antigen, and another part is bind with the other cells. Another part bind with the, you can say, another immune cells, and thereby there is a microbial destruction. Or you can say that phagocytosis. Fine. Now it means that antibody IgG work as a opsin, and the process is called as the opsonization. Clear? You want me to repeat?
Yes, sir.
Yes. So opsonization is the process by which antibodies, one part bind with the antigen, another part bind with the other immune cells and facilitate the microbial destruction process or phagocytosis. It means antibody IgG behave as a opsin, and the process is called as the opsonization. Clear? Yes? Understanding? Yes? Fine. So this is all about the IgG, and you should remember one more thing, that it do have the subclasses. It do have the further some subclasses like IgG1, IgG2, and like IgG3 and IgG4. Fine. So let's recap what we studied about the antibody IgG. Antibody IgG, you just remember, okay, concentrate here. IgG, you just remember, it is the first most abundant, fine, and a large molecule. Weight wise, it is a 150 Dalton, fine. Life cycle is 23 days, and it's also help in their what is called as the why they are most abundant, fine. In the whenever microbes comes in our body, there will be a first immune response, and the main antibody which produces is the IgG. But in the secondary response, there will be the IgM. Now, this exists in a monomeric form, single form, fine, and it can cross the placenta. And it will help in the opsonization, or you can say it's perform the opsonization, means basically help the other immune cells to, you can say, destruct the microbes antigen. And the thing is, it exists, they have the subclasses like IG1, IG2, IG3 and IG4. Clear? Yes? Shall we move to the next antibody?
Yes, sir.
Fine. So your next antibody is IgA. Next antibody is the IgA. See, I am teaching you bit detail just because there is one exam in your nursing, that is called as the NEET. Have you heard about that?
Yes, sir.
Yeah, for being a nursing officer in the AIMS, a prestigious, you can say, the colleges or hospital in India, that is called as the AIMS. So in that, you can come across these questions in your for MCQ dealing. If you studied in this semester, no, probably you will not study the same microbiology in your next semester. But whenever you have the note, whenever you revise, you will recall. Okay. What INC nahi hai? Why you are worried about INC? It's a college matter. They will take care. Don't think about this. Study on your focus on your study. That is more important. तुम उसके सोचने में लग जाओगे, तुम्हारा 4 साल ऐसे बीत जाएगा, पता ही नहीं चलेगा. You will got the degree, no, that is enough for you can say competing in any exam. No issue. Focus on the study more. Okay. Next antibody you have is the IgA. The thing which I said you is that this IgA, it is the first thing, is it is the second most, it is the second most abundant. Another thing which you have to understand is that this exists in a two form, monomeric as well as the pentameric. When it is in the, you can say, when it is present in the blood, okay, when it is present in the blood, it is existed as a monomer form. When it is present in the blood, it exists as a monomer. But when it is present in the mucosal linings, it is present in a dimeric form. And its life cycle is 6 to 8 days. 6 to 8 days. See, first thing I said that it is present in a monomeric form when it is in your blood, but when it is in a membrane, mucosal membrane, it exists as a dimeric form. See, you know that you have the respiratory system. Yes, you have the respiratory system. You have the respiratory system, and the thing is, the internal lining is called as the mucosa, isn't it? It's called as the mucosa, and this mucosa, no, it is having the, yes, it is having what? IgA antibody. IgA antibody. Not only on the respiratory mucosa, your very much GIT, your GIT, it do have mucosal lining, and this mucosal lining is basically having the antibody IgA. It means that accidentally or somehow, if the antigens will comes in your respiratory tract or your GIT tract or your urinogenital tract, fine, there must be any protection? No. So that protection is mediated by the antibody IgA. IgA will start, you can say, fighting with the antigen which comes into the GIT tract or respiratory tract. Clear? Yes. So it is acting as a antiseptic paint. Antiseptic paint over the mucosal lining. Just write it down. It's act as a antiseptic paint on your mucosal lining. On your mucosal lining. Clear? It's also help in the phagocytosis. It's also help in the phagocytosis. Clear? No problem. Then the breast milk. Okay? The breast milk is having the antibody IgA. Breast milk contain the antibody IgA, which basically protect the newborn, Clear? Yes. What else? And it's help in the phagocytosis, you retain. Then their life cycle is 6 to 8 days, so it is a short-lived basically, and almost all the body secretion, not only the breast milk, all, as I said, you know that your tears is acting as the antimicrobial in nature, because in the tears also the antibody which is present is the IgA. Apart from that, you having the saliva in your mouth. In the saliva also there is a presence of IgA antibody. So hence you can make one statement that most of the body fluids, okay, most of the body fluids which is secreted, that is having the antibody IgA. Apart from that, all the mucosal lining is having the antibody IgA. You can remember easily IgA and A for antiseptic, A for IgA. Okay, IgA antiseptic paint. Clear? No problem. Now let's move to the another antibody called as the IgM. You have any doubt till here?
इसका blood में life उतना ही रहेगा।
हाँ, 6 to 8 days only. They are short lived. 6 to 8 days के बाद मर जाएगा, इधर एंटीबॉडी फिर नया बनेगा. It's a replacing. Okay, replacing old by the new antibody. Now coming to the IgM. So you have antibody IgM. What we learned? We learned that there is a, yes, very good. This comes in the secondary immune response. First immune response is a small one, IgG. Another immune response, is the IgM. Okay, so write it down. This is the antibody of secondary response. This is the antibody of secondary response. Written?
Yes, sir.
Then you write it down. It exists in the pentameric form. It exists in the first one is it is a antibody of secondary immune response. Okay. Second thing which you have to understand is it exists in the pentameric form. Okay, it perform one reaction, basically the two reaction. The first one is called as the agglutination. Okay, another one is called as the precipitation. These terminology you can come across in 2 marks. Okay, see, IgM exists in the pentameric form. Fine. Now, what mean by the pentameric form? Pentameric form, there will be a one J chain which join basically. So there is suppose this is the J chain, and here the 5 antibodies join. 1, 2, 3, 4, 5. So 5 antibodies join on a J chain. Fine, they constitute the pentameric form of antibody. The thing is, these antibodies, they will react with the RBC membrane. Okay, these antibodies, they will react over the RBC membrane, and you know that RBC is like a biconcave in shape. Yes, RBC is a biconcave in shape, a disc shape, basically hollow disc shape. Okay, and suppose there is an antigen which is present over the RBC membrane, then our antibody will react with it. And there will be a RBC clumping. Okay, RBC का इकट्ठा होना, clumping. This reaction is called as the agglutination. Agglutination. Fine. Yes. Mainly IgM will protect our blood. Mainly it protect our blood. Fine. Mainly it protect our blood. So whatever you can say the blood-borne infections, whatever infections comes in the blood, this antibody will actively participate and destroy the pathogens or microbes which is present in the blood. Fine. Now, the precipitation reaction means when you can take, this is what is called as the test tube. Fine. And here there is the antibody. So when this you provide the IgM, okay, अगर तुम इसमें IgM को डालते हो तो ये जितने भी छोटे छोटे एंटीजन है ये सबको नीचे सेटल डाउन कर देगा, प्रेसिपिटेट करना, प्रेसिपिटेशन रिएक्शन. Clear? Yes, IgM done. No opsonization here, no phagocytosis here. Fine. The thing which is happening is the agglutination and precipitation. Clear? Okay. Now let's move to the another antibodies called as the IgE. Yes. इसका life cycle is they live very short. You can say 5 days. Okay, so they live in the 5 days. They exist in a 5 days. Yes, 5 days. Fine. Now, talking about another antibody called as the IgE. IgE, it is basically formed whenever you have the allergy. So whenever you have allergy, the principal antibody which is formed is the Yes, IgE. So it is basically found mainly during the allergy. We will discuss what is allergy in few minutes in the hypersensitivity reaction. Fine. Half-life is 2 days. Half-life is 2 days. Mainly it is mainly the number goes high during the allergy, like asthma. Okay. Asthma, something called as the hay fever. Hay fever. So in these diseases, the number goes high, and the thing is, these antibody, they love to fit over the mast cell. They love to fit over the mast cell. Mast cell is basically one specialized form of B lymphocyte only. Okay, this is your antibodies, and it's love to fit over the mast cell. This is the mast cell. So when antigen will bind over here, when antigen will bind to this IgE antibody, it's inform the mast cell, and mast cell will secrete something which is called as the spasmogenic as well as the spasmogenic substances. It will generate the spasmogenic substances like prostaglandins, These prostaglandins will start contracting. It will start the contracting, and when there will be a, you can say, more exaggerated response, then this mast cell, it will release another chemical mediators like leukotrienes. Okay, these are the chemical mediators, small chemical mediators, which basically start inflammation. Inflammation is the protective mechanism for our body. Okay, so these things triggers the inflammations, and thereby they will destroy the microbial antigen. Okay, so there is a prostaglandin secretion, then there is a leukotriene secretion. Fine. Apart from that, this mast cell, it releases the histamine. It releases the histamines. Fine. So these are the spasmogenic substances. These are the spasmogenic substances. Fine. Clear? Once again, no? Okay. Wait a while. Spasmogenic, and there is one more thing.
तो मास्ट सेल जो को कुछ रिलीज करता है तो वो लिखे और एक और क्या नाम है?
Vasogenic. Yeah. See, the thing is, as I said you, IgE antibody is mostly produced during the allergy. Whenever allergens come, suppose you inhale the pollen grains, okay, or any dust, or any kind of, you can say, the another food, even the food, someone is allergic from the food. So whatever gets inside your body, it will that is act like antigen. So antibody must be produced. No, so the antibody which is produced is the IgE. Fine. The thing is, this IgG is basically membrane bounded. It is a membrane bounded antibody, and the antibody which bind over which cell's membrane is the mast cell. Mast cell is basically a specialized form of your B cell, B lymphocyte. It is a B lymphocyte, but differentiation make it mast cells. Fine. The speciality of this mast cell is that कि जब कभी इसके ऊपर में एंटीबॉडी बाइंड रहेगा और एंटीजन जब बाइंड करेगा, तो ये एंटीबॉडी ट्रिगर करेगा इस मास्ट सेल को. ये ऊपर में प्रेजेंट है सेल के, बट अंदर में इंट्रासेलुलर सिग्नलिंग आएगा. मास्ट सेल क्या करेगा, तो कुछ स्पास्मोजेनिक एंड वेजोजेनिक सब्सटेंस रिलीज करेगा. स्पास्मोजेनिक मीन्स ये कॉन्ट्रैक्ट करवाता है. सपोज इट इज प्रेजेंट इन द ब्लड वेसल, देन इट विल कॉन्ट्रैक्ट द ब्लड वेसल. ओके, ब्लड वेसल कॉन्ट्रैक्ट करके वहाँ पे ब्लड सप्लाई लेस कर देगा. and these things triggers the inflammation. तो जो वेस्मोजेनिक और स्पास्मोजेनिक सब्सटेंस रिलीज करता है, पहला सब्सटेंस है प्रोस्टाग्लैंडियंस. अनदर सब्सटेंस इज कॉल्ड एज द ल्यूकोट्रिन्स. ल्यूकोट्रिन्स, ठीक. और एक सब्सटेंस रिलीज होता है जिसे हम लोग बोलते हैं हिस्टामीन. हिस्टामीन, ये छोटे छोटे केमिकल्स हैं जो मास्ट सेल रिलीज करते हैं, और ये केमिकल्स के कारण क्या होता है बॉडी के अंदर में, तो कॉन्ट्रैक्शन होता है ब्लड वेसल्स का, ठीक है. स्पास्मोजेनिक एंड वेसोजेनिक रिएक्शंस होते हैं, जिसके कारण क्या होता है, तो इन्फ्लामेशन होता है. व्हाट हैपेंड इज इन्फ्लामेशन. क्लियर? यस. You have any doubt? Anyone? Clear? समझ आ रहा है क्या आप लोगों को? पक्का? Fine. Then we'll move to the next class called as the IgD. About the IgD antibody, you just remember it is a less, in trices it is present only, the amount is very very less. Okay, it is a least one, it's the least antibody. Another thing which you have to understand is that it is present over the B cell, it is present on the B cell, exist in the monomeric form, and life cycle is 3 days. Okay, we don't know much about the IgD antibody, the very much description is there, so हम लोग को ज़्यादा डिटेल में इसको पढ़ना भी नहीं है, क्योंकि इसके बारे में ऑलरेडी बहुत कम इंफॉर्मेशन अभी तक है. The antibody is called as the IgD. It exists in the monomeric form. It exists in the form. Got it? Yes. So this is all your 5 classes of antibody. Do you have any question? Yes. You have any question? No. So let's conclude the class about the antibody. What we discussed. We discussed that antibody is it's a glycoprotein which basically act on the antigen, recognize the antigen, act on the antigen, and either facilitate or destroy the antigen. Then we discussed that the structure of the antibody, a heavy chain and light chain, differentiation is on the basis of their weight. We discussed the specific part in the antibody. There some part is a constant part, some is a variable part, some is specifically meant for antigen binding. Okay. We discussed the what is called as the valency, avidity, okay, affinity of antibody. We discussed the forms like monomeric, dimeric, and pentameric. Then we also discussed about the, yes, what else we discussed? We discussed the antibody type, and in today's class we discussed the antibody type in detail, starting with the IgA most abundant, then IgA, IgM, IgE, and IgD. We discussed their different forms where they exist, their action we discussed. Okay, so this concludes your antibody lecture. Fine. Now we'll move ahead and we'll discuss the hypersensitivity reaction.